disease transmission, and the motivation is like, as I said just now, it is based on the annual review paper mostly about the fluid dynamics and the disease transmission by Lydia Bolivar. So, like there will be many like figures. Most figures is not written. Anything will be from that review paper only. Before that, like the motivation, as you all know, that we all encountered COVID-19, which was like highly communicable disease. Before that, also many times in the East Asia. So this is severe acute respiratory respiratory syndrome, right? SARS-CoV. Mars is the Middle East respiratory syndrome. Then tuberculosis. All these are airborne diseases. Then, like we all said, like we all have knew like how 6 feet or 2 meter, like the physical distance maintenance on what is the basis, and then are not sufficient for protection. Many times also there are discussions about like the crowded places in hospital rooms, transport system floors, how much or in what is the effective way of its decontamination. Also, in case of plants, like few last few slides were about the disease transmission through like droplet interaction of these leaves, how that can also happen. So here are like a schematic of the four stages of any disease transmission. So the first part is more like some biological terms. The first one is like extraction and encapsulation. We did not bother about that, but you can see here the fluid dynamics part involves complex and viscous fluids rheology, which involves with the characterization of the polymeric fluids. Then hydrodynamic instability is there. Again, for disease detection, we need some sort of lab on a chip kind of diagnostic tools, which involves our knowledge of microfluidics, surface fluid pathogen interface, how the fluid like contaminates the surface. That can depend upon the wettability of the surface, whether it is hydrophilic or super hydrophobic. And then like the pathogen fluid interactions that can also maybe like sometimes either the droplets become into smaller droplets or the bubbles they can burst and they can spread the pathogens. Similarly, although we don't know anything about symptomatology, you can see so many like hydrogen phenomena is written there. Just like that, pathogen physiology here also phase change, interfacial flows, adhesion, absorption, the fluid filled droplet, these sort of microphysics is involved here. Here also, like pathogen penetration aerodynamics in the last stage when the exposure and infection is taking place. So as you can see, the first part is will be done by the doctors and the biologists. In the second box, here we have some expertise. Like most of us are either mechanical or chemical engineers. There could have been applied mathematicians also, like Manoj sir and his students. So everyone can chip in the second one. So these are the different stages in each different physiological stage. All the aspects of fluid dynamics is tabulated here. Some day-to-day events, like one of the, like this is a kind of particle tracking of the curve staging. You can see comparison of the tracks detected from high-speed trains using the particle tracking velocity plate. So basically, we will track the of the droplets and the speed and how much it gets like spread out. So here coughs or sneezes, spread of droplets from bursting bubbles, crashes from wet contaminated surfaces. In the next figure, you can see here. So this is like a image of a person. You can see this is the mouth through which like someone is coughing or sneezing. That figure. Here, the figure B, like as I was saying, like let's say some leaf is there, some infected kind of virus which can like affect a plant. Let's say some droplet is like falling here, then this is a again a flexible surface. It like kind of bounces it back, it spreads to another plant, and in that way, plant can also spread some like virus affected or pathogen affected droplets here. This is like one of the like the high speed jets when one passes the toilet, someone has taken photo, and so this comes basically in the form of jets, and also it breaks in the form of like smaller droplets that can stay airborne. This is like let's say a bubble which is eventually bursting up. and releasing, you can see very small droplets here. So these are the instances which you can readily relate. So in context of like when we talk or sneeze, there are some observations like isolated drop or like the collection of drop. So here the respiratory drops are like basically the drops which we release in the form of coughs, they are in the range of 1 micron to 1000 micron. 1 milliliter is equal to 1000 micron. Then it can happen that it drops on the floor and it eventually evaporates. That solid residue itself can be like responsible for the like the disease transmission. And this solid residue after the evaporation, this is called droplet weakly. This is just a, like a jargon, and these are in the order of 10 micron. And these first sort of how these sort of droplets evaporate and how much they stay airborne, this was first studied by this scientist called Wells longer in the first half of the 20th century when he was studying the how the TB it transmits. So he measured the evaporation time as well as the settling time, but he observed that when the droplets are bigger than 100 micron, you can see that it basically within 1 second from the moment we release, it settles down. So these the setting it can be like readily seen. Like so, you can start you already setting velocity. So I just show you here. You can see, as I said, that the larger the particle diameter, the p square is basically the droplet diameter. So bigger the diameter, high is the settling velocity, and it will quickly kind of settle down to the ground. And the drops smaller than 100 micron typically stay airborne and try to vaporize. So these small droplets, when it vaporizes, you can see then only the solid residues goes up a droplet mainly. But
one great thing, so
so you can sit in the past because I have a lot of knowledge here. He can like really compliment. Like that.
one great thing, so in our derivations we assume that rho p is much better than rho f. So if you connect that, that rho f is not there.
So rho F is basically the air density is equal, like, it is considered it is much higher. So that's why like rho F is negative.
But this is the general formula.
Yeah, you can see that D square essentially like decides that the large drops that can like settle down quickly. Now, again, there is another paper which was like more than 100 years. It's Kluge, 1897. He showed that just like we said that the larger droplets it like settles down within 1 second. He basically calculated what is the distance along which the larger droplets settle down. So that is 1 to 2 meters. And then the smaller droplets, it stays airborne. Just in case, many times it has been shown recently, like by CFD analysis or like experts also, whenever like let's say this sort of indoor, if there is some AC going on or some ventilation system with ambient air current, these airborne smaller droplets it basically transmits much farther and like it can travel quite a distance. Now, so based on that, it's like 1 to 2 meter. Like 1 meter is roughly 3 feet. So the 2 meter or 6 feet, we like do or some whatever some Hindi slogan was introduced during COVID. That was like it was based on this slogan. Like, so it was long back. It was like already observed. So that kind of empirical safe distance is given here. And so one thing it was written that like the impact whether the large droplets are mainly responsible or whether the smaller, more long time airborne smaller droplets are responsible, that is individually like whose contribution is more important, that is kind of difficult. So the WHO or the regulatory body they didn't say anything particular that which droplets are more responsible. But as you can see in the mask design, like first we used to wear the normal the dispensary mask. Then later we were told to like wear the N95 mask. So basically the N95 mask, I guess they can screen the more smaller droplets. I guess that is the reason. Okay. So Then, like here is the curve droplet, and when someone tries to model it, these are the initial parameters: volume, density, and the buoyancy. The ambient density is rho a, that is the air, gravity, and the momentum. So, one tried to model, it has been observed that it is like a kind of jet or thermal plume, and it is expanding like in a conical form. We'll come to that. So you can see here that it is still. So yeah, some of these was some. So this is called jet interface. You can see basically we release some sort of fluid, it gradually entraps the surrounding air. So there is, you can see laterally this entrapping some air, the fluid momentum basically it becomes much more wider. So that has been only like just schematically described here. And here you can see the WT is basically this W, the lateral velocity, is described as alpha of ut. If ut is the axial velocity, alpha is the entrainment coefficient. Again here you can see that when one started the droplet, like the dynamics of the transmission of these curved droplets, it can be seen that the larger droplets they quickly fall down, and based on the distance till which the larger particles are airborne, you can see from her mouth to the this you can see this is falling along the trajectory, and this axis is called the range of contamination. The range of contamination basically that kind of determines the safe distance from which one person and another person should talk or whatever, like any sort of interaction should be maintained keeping this physical distance. Now, this sort of jet, as I said, this entrapment, like if you look, it's basically from the Stephen Pope's chapter 5, it's about the three shear layers, like it's mostly about the turbulent jets. So you can see here, this is a velocity profile, which is like very near to the mouth, or in context of biological systems, you can say a point of acceleration. So this is x by t 30, this is x by t 60. It means it has progressed further. So the maximum velocity has reduced. It has, as you can see, the width has increased. As it has crossed x by t to 100, the maximum axial velocity has decreased, but the width has been like increased accordingly. And then the shape we have called like the jet entrapment, it's a self-similar behavior because just like when we have studied the 2D flat plate boundary layer, we have studied the glacier similarity solution, right? So in the glacier similarity solution, what we do, we do the like u by u infinity and plot y by u, and then we say that at all locations, if we non-dimensionalize, the velocity profiles will match. Likewise, here also, if we like do the u by u0, u0 is going to be the initial maximum velocity by the distance, the central, this one, like the axial distance by the initial, the r half, that is the one half of the central jet initially, then it looks like this. So it essentially overlaps on one another. That's why it is called shape similarity. So this turbulent gas cloud, which we are saying like the coughing or sneezing, in general, if we don't like keep in mind like the biological aspects, these are known as turbulent puffs or jets. In context of geophysical flows, let's say some sort of volcanic eruption is happening, so that those are called turbulent fumes, or these like let's say in the seabed, whatever sometimes the geothermal vents, like some sort of like holes are there in the seabed through which the hot gases are emerging out. Those are called thermals and plumes. And here you can see the thermals and plumes are also driven by puffs and jets and momentum driven. So let's say like if we study like whatever is the dynamics of the like determined like nozzle after the combustion. So see we will know like going after the combustion through a nozzle, whatever is the exhaust, it releases in the form of jet. So those features, if we study those features, the observation of self-similarity and the entrainment hypothesis observed there can be applied while modeling the Half J also. Yeah, so then we said here the trajectory of the axial droplets are analogy with geophysical flows with riverine outflows. So let's say some sort of like riverine outflows is basically like let's say a river in the delta region is entering in the ocean. The way it like initially enters like with a large velocity, but eventually it kind of diffuses out or the industrial wastage, like from industry, let's say any oil refinery, and when it dumps in the river, such sort of large volume it initially comes in the form of jet, and eventually it kind of disperses out. So those features will be like these biological flows. So these long sentences will did not bother, like there's some quantities like the air volume emitted, you can see 500 ml for regular accelerations, but when we like impart momentum while curves or squeezes, it changes to, it has increased to 1 to 4 liter. Likewise, I don't know what is tidal breathing, you know?
Tidal breathing, yeah. So tidal breathing is basically a measure of the inhalation rate. In how it is measured, if I remember correctly, it is probably, yeah, let's not say something which is inaccurate on video, but yeah, it is a form of measuring the inhaled air flux.
Okay.
That is so basically it should be the inhalation rate that we have been using multiplied by some factor.
Okay, so you can see for a tidal breathing, and then when we compare the curve and the sneeze, the duration of the emission has changed. Likewise,
One quick thing, that point where we introduced the populations for the breeding rate, remember for the adults and for adults and other females. So that paper that I cited over there, if you are interested, you can go into that paper. They do go through all the definitions for that.
yeah, so you can see here the Reynolds number for normal breathing is 10 to the power 3, but since we are inverting much higher speed during coughs or sneezes, you can see the Reynolds number has increased by 1 order magnitude, that is in the order of 20 to the power.
So the last sentence you see normal bracket tidal breathing. So
Okay, so it's maybe like the relaxed breathing. Okay, yeah. So you can see here that all the parameters compared to the normal and the whenever the high momentum is involved, those are quite different from each other. So these sort of physical quantities one have to remember while modeling the like when you are applying simulation features. So now, like, what is the fragmentation of mucocerebral fluid? So sometimes, like, we know that our cough contains mucus. Once it releases from our mouth, how does it break down? So you can see occasionally a mucus contains like mucin, some sort of biopolymers, protein salts, and moreover, due to the presence of these biopolymers, it is viscoelastic in nature. So the fragmentation is also important. So these are the side views and the top views. The top being released from the mouth. Some high-speed imaging has taken place. So you can see occasional long filaments have come during the breaking process. And these long filaments, as you can see, it's observed also in our group. Some of us have done. So these are called BOS structures, boas or beads on the string. Occasionally, it will be like beads, like long filaments in between some sort of bead structure. So it's as if like the beads are interspersed between the thin filaments of liquids. That's why it's called beads on the string structure. And we also know that compared to water, whenever there is polymer due to the fluid elasticity increase, the beads, these BOS structures will appear, and these ligaments will take much longer time compared to the water filaments. So here, obviously, the breakage of these liquid filaments, these are governed by the interfacing mechanics, and many of us know the when we involve surface tensor, there is Weber number which is rho b square by b square a by sigma that kind of takes care of the competition between inertial force by the interfacial force. Similarly, like in any interfacial force, instead of Reynolds number, we take into account the Weber number to take into account the competition between inertial and the interfacial force. But let's say we also want to change the different kind of like fluids with varying viscosity. So then, horizontal number, you can see It is the ratio of viscous force by inertial force into interfacial force. So the formula is this, as you can see, it can be also expressed as square root of Weber number by the Reynolds number. Bond number on the other hand, like we have, like from the very first of our slides, we have shown that in curved droplets, there are like some droplets are in the order of millimeter, some droplets are in the order of like micron. So what we know that larger the droplet, obviously the gravitational effect will be stronger. So here the bond number basically it tackles the ratio of gravitational force by interfacial force. So when bond number is much greater than 1, you can see that larger the diameter of the drop, the d square term becomes dominant, and then the effect of gravitation is naturally higher compared to the interfacial force. So and these can be also like expressed as two length scales. So here this is d square. We take down sigma by rho g. The square root of sigma by rho g is the capillary length scale. So capillary length scale, and for water and temperature around 20 or 30 degrees Celsius, for like mostly we find that it is around 2.5 mm is the capillary length scale. So these bond numbers are also important while we take into account the path dynamics or the fragmentation of these sheet dynamics here. Yeah, so again, just like the capillary length scale, capillary time scale is defined as the square root of rho d2 by sigma. So basically, like let's say if we release a free droplet, the oscillation, like we can see that the droplets kind of change its shape while it falls down. The time scale of the change is governed by the surface tension as well as obviously the diameter of the droplet is there. So this is a combination you can find out like by calculating putting the dimensions of this one, it will eventually find the unit is in time like of second. Okay, so here you can see lowering the surface tension will enhance fragmentation into more or smaller droplets. So here this is a fact, like if we like if add surfactant to the base water, it will be easier to form smaller droplets. So basically like in order to fragment, what does it mean? Like it essentially enhances the area, then it breaks up. So that requires some energy, right? If you want to expand the interfacial area, it requires like interfacial area into A, sigma into dA. So what to be done is sigma dA. So the point is that if we reduce the sigma, the work requires less. So that's why it is written that for easier fragmentation, if we add surfactants, it will be more easily breaking up and releasing smaller droplets. Likewise, another feature, like just a little bit of trivia about the viewer structures, you can see. So these are some figure from the Gareth McNeil's group, published in Nature Physics 2010. You can see it later. So these, like let's say, the saliva in our mouth, someone has tried to do some sort of like stretching between his hands, and you can see that it has formed some sort of filaments here. Likewise, some water soluble polymer, it can be elongated. So these sort of like if you try to do it with water, this sort of like long lasting filaments will never appear. Again, here you can see the satellite beads are appearing. Even further, it is termed as sub-satellite beads. So these are the features of beads on a string kind of thing. In our group also, we can, like we have seen, like when we have dropped this 1500 polymer concentration on a superheated surface, not exactly beads on a string structure, but some sort of liquid filaments are appearing here. So you can see during the refraction process, thin filaments are being formed, and these are like long lasting enough to cool down the bigger part of the droplet. In case of water, during the refraction process, first of all, these filaments, even if it forms, it will last very few seconds and it will break up. The water will like just get evaporated. On the other hand, with same set of polymer concentration, you can see in the inclined plane that let's say droplet is falling down of same polymer concentration, you can see how much it gets stretched. Okay. This is 60 degree, but you can see the stress like with a much powerful lens. You could have seen a like a actual black line here due to the lighting and due to the like lack of magnification. You can see the proper like thin ligament will form. These filaments are essentially features of any non-Newtonian drop impact dynamics. So these are observed by Somo, who has passed, and Khusro also has seen such kind of things. So here is again just Khusro's recent paper. He has shown that you can see that this is like the effect of Weber number on the diffraction dynamics of 2000 ppm drum on the NCZ surface. NCZ is a different kind of superhydraulic surface. You can see here that at the lowest Weber number, it just drops here. This is the top view of the dynamic. So as we increase the drop rate, this is the ring, and from the ring, what happens in case of water, it basically due to the ring instability, the rings get basically some sort of finger-like structure happens, and in case of polymers, some sort of small bead-like structures appear here. But non-Newtonian fluids have sufficient elasticity. In case of water, these sort of thin filaments they fragment, and then you can see secondary droplets are breaking away from the main droplet. Here, what is happening that due to the fluid elasticity, these thin filaments are always intact throughout the refraction process. This never breaks, and eventually again comes back. And this is a paper by senior Somo. What he has done is simply like allow the non-Newtonian drop to fall on the cylindrical surface. For a cylindrical surface, since the observation time is much longer, you can see the what happens in case of water. Like let's say this is cylinder, the droplet when it falls down like this, it stretches, but due to we know the capillary breakup is governed by Rayleigh-Plateau instability. So the Rayleigh-Plateau instability, like within 4 to 5 milliseconds, these long filaments will break up. But in case of polymer, it gets stressed and stressed. We know that in the non-Newtonian fluids, the extensional viscosity is much higher compared to the shear viscosity. That keeps these long stretched filaments sufficiently long lasting, and you can see the emergence of small waves. So, although like in a different context you study the drop dynamics, what I am saying, the ligaments which are appearing in the top is can be also observed in this kind of droplet dynamics here in case of the non-Newtonian flows. So now when we discuss the non-Newtonian flows, there are two relevant non-dimensional number. One is the Weisenberg number. Weisenberg number is the ratio of the polymer relaxation time. By polymer relaxation time, we mean that the polymer, if we can imagine some sort of spring-like structure, initially they are coiled under high shear, they stretch. So the time scale over which the stretching takes place and again it gets back to its coil or the equilibrium state, that statistically average time is called the relaxation time by the flow time scale. The flow time scale, sorry, this should be lambda gamma dot. So it is basically Weisenberg number equal to lambda 1 over gamma dot. So this becomes lambda gamma dot. So that is a wrongly typed. So this is important, this number. Whenever the Weisenberg number becomes greater than 1, this means that the non-Newtonian features will become sufficiently strong, like whatever let's say we are saying elastic fluid. And when the fluid is like moving at a very low velocity, the polymers are not stretched enough. The fluid is not able to exhibit its elastic property. So then, Weisenberg number greater than 1, it implies onset of elastic instability. In case of pipe flows, unless we exceed the Weisenberg number greater than 1, we can't see the drag reduction. By drag reduction, again, this is slight digression from the disease transmission, but since we are discussing anyway, so earlier we said that this is the laminar flow, this is the laminar flow 64 by Re, and this is the 0.3164 Re to the power minus 1/4, scaling this for turbulent. If we use polymers in the turbulent state, in presence of polymers, the friction factor may be somewhat lying in the intermediate zone. So let's say at a Reynolds number 10 to the power 4, 10,000, the normal Reynolds number friction factor is here. If we mix some sort of polyethylene oxide or TAN, this friction factor will be lying in between. So that is what happens if the Weisenberg number exceeds 1. Now, just like the Weisenberg number, many people prefer the elasticity number. Elasticity number is the ratio of Weisenberg number by Reynolds number, and by these two ratios, we eventually cancel out the fluid velocity, and this quantity is like a collection of all the material properties. This doesn't include, you can see, this is the numerator is dependent upon the relaxation time and the viscosity. So, let's say the viscosity of two fluids is same. If we increase the lambda, that will increase the elastic number. So, higher the elastic number, higher is the elasticity property of the fluid. Likewise, let's say sometimes people say that we want to increase the elasticity number. So, we mix like 1000 ppm in water, and on the other hand, if we mix like let's say water and glycerol mixture, and then we mix the polymer, the viscosity of the water glycerol mixture is obviously much higher compared to the water. So in that case, the elasticity number will be obviously higher compared to the water plus TAN solution. Okay, so we are discussing the fragmentation. Regarding the fragmentation part, there are many sort of hydrodynamic instabilities also involved. Hydrodynamic instability itself is like a one semester or two semester course involving long equations. But here, just to cover the responsible hydrodynamic instabilities in the fragmentation process is like the first one is Kelvin-Helmholtz instability. So basically, this happens when there is a difference between two fluid interfaces. So here, let's say initially there is some white cloud, and this is moving at some distance. The wind over it is moving at another distance. Due to the interfacial velocity difference, there will be Kelvin-Helmholtz instability appearing in the flow. Likewise, as I said just few minutes back, that the BOS structures can like withstand the onslaught of the electrical instability, but this is what is happening in case of a water jet. So the water jet, basically, these are also called capillary breakup, and you can read this about in much detail. There are, like this is the review paper, but if you just want to get a basic idea about what is happening in instability, you can read this book by either Resin, DRAZIN, or Resin and Reid. So there the details of this dynamic instability or any sort of instability, you can get an idea. But this has to do with the surface tension. Essentially, what it does, the surface tension, it always follows the principle of minima, like principle of what? Area minimization. It always tends to like whatever the interface is being formed, the orientation is such that it will lead to minimum energy configuration, and that is like once we achieve the minimum area, that will lead automatically to the minimum energy configuration also. Similarly, like the Rayleigh instability, there is another sort of instability, the Rayleigh-Taylor instability, when, like you can see, when the lighter fluid is pushing the heavier fluid. So let's say initially, like we create a system where, like this is oil, this is water. The water is heavier than oil. So essentially, since it is an unstable system, the water will tend to push through the oil, right? So that creates this sort of instability. So what the this review paper, Eggers and Villermo, they like kind of suggested that the fragmentation process is obviously a complicated process, and this is a combination of all these three hydrodynamic instability process. So much more you can read it individually about the hydrodynamic instability regimes. Yeah. So this is new term. It's not a full screen. So this term, yeah. So this nosocomial pathogen transmission, by definition, it means that the pathogens which are transmitted by the respiratory route, sorry, means other than any respiratory route, like if there is, like, let's say, other than like air transmission direct, if these kind of things happen, then let's say drop some sort of like a long jet or the splashes from surfaces of the toilet flushes, basically the water impacts the surface, then it releases the smaller droplets in the air. So here, like the splashes from the surfaces, the relevant fragmentation process which we just discussed, and the aerosolization, basically like the smaller droplet released in the air, the whole like process here the relevant Weber number is 10 to the power 3 to 10 to the power 4. So this figure you showed, like we showed earlier, is high speed imaging of droplet trajectories emitted from a high pressure flush of a typical hospital toilet. So essentially someone actually like made the hypothesis. I think the writer herself and the student, like obviously not going to the bathroom, made some sort of setup and then did the class, and then they studied like if we release some sort of high speed water in the toilet flush like setup, how will the reflected droplet trajectories look like? And they said that from the droplet trajectories, this can rise quite high, and this sort of liquid jets has a high tendency to break up into much micron level smaller droplets.
So one thing over here, so the word nosocomial does not come from nasal. It is
Yeah.
related to anything that is happening in a hospital setting. So because there, whoever, if you have patients there waiting for injuries that might be already pathogenic in nature, so nosocomial transmission is pretty common, and there is a good body of literature on that as well.
So this is like seeing the droplet trajectories and the subsequent release of the smaller droplets. They have studied how far the these, you can see these dotted lines are basically the jets ejecting out from the flask. You can see the relative fluid volume fraction. So they have kind of some sort of made some contour map of the fluid released.
That's why my wife asked me to close the lid all the time.
Okay. Yeah, yeah. So like when this paper came, then like people actually made this sort of conscious decision. Like because it was COVID times, everyone was in the, like unless we have life threatening, we don't follow the good practices. That is sad but true. So like you can see that this has to like be like leading with the slashing dynamic. Here, like I will show some case, like one of your group mates, Mangles, has studied like how the splashing dynamics is affected by the presence of polymer droplets. So here you can see that let's say what he did, like made a simple water film, and then like he made some different kind of solutions having different polymer concentration. So here you can see that yes, like change the polymer concentration of this is ranging from 250 ppm to 2000 ppm. But it shows like at the low polymer concentration, the splashing effect, like in just water, these sort of small thin structures, they will readily fragment out. Here at 250 ppm, you can see the filaments eventually gets broken, the fragmentation is taking place. But as we increase the polymer concentration, these filaments are quite robust. They will always stay intact, and eventually, if we follow the whole experimental time frame, the filaments will eventually merge with the water. So basically, splashing is inhibited in these sort of scenarios. In the next slide, this is again magnet only. So the first phase is water droplet on water film. The second droplet is viscoelastic droplet on water film. The third one is viscoelastic droplet on same viscoelastic film. Like the video is made in such a way that it will play at the same time. So before and I am telling. So this will, you can see here fragmentation will happen. Here, like there will be the rim instability and the crown formation will take place, but the secondary droplets which are formed at the tip of the filaments will stay. Here, the crown will not even form. It's like crown will form, but the secondary structures coming out from the rim, those will be not formed. Okay, so it is not playing like this. Let me just play it individually. So let's just repeat one more time. So you can see in the first figure it will break. The second one there will be secondary structures. The third one there will be not even filaments coming out from the outside rim. So you can see right in the water case the secondary droplets are breaking away. This one viscoelastic droplet is water film. There is rim instability happening. But when it is viscoelastic droplet falling down on the viscoelastic film, there is no rim instability then. Okay. Now, water to air transfer and bubble. So we said that bubble bursting is also a prime mode of air transmission. What happens? Like let's say the bubble, the thin liquid film may contain pathogens. It can burst and release the virus or the pathogens in the air. So here, slided imaging is a form of technique. To the slided imaging, you can see a bubble and these small dots are the bacteria. So the slided imaging of bacteria that is bubble. And these will stay as long as it is there. And it will not die out. So here the evaporation process is also an important thing to study. Now, many times it happens, like let's say some bubble is resting on a solid surface. What happens? Because there is a spherical, like this curved surface, due to the gravitation effect, there will be drainage of the film downward side. And eventually, there is maybe some sort of hole formation is taking place, the bubble will burst. So these are the different stages of the bubble bursting that is captured here. So you can see eventually, like first initially, there is some hole formation here, and as the hole like kind of spreading rapidly, there is emergence of the secondary droplets on the hemispherical cap, and ultimately it is bursting out.
The one the one quick second over here. So this is relevant for transmission, right? Now can you guess, as if you think about someone who is infected, how is this phenomenon relevant for the pathogen from that person's body to go out into the air? Think about when someone is talking. We are closing our lips and opening it. It's like a bubble like a film with cracks, bursts, and then it can create a particular surface.
So bubbles you know from the Young Laplace equation, there is the delta P equal to 2 sigma by r. This can be readily explained if you know the Young Laplace equation, which is delta P equal to sigma 1 by r1 plus 1 by r2. For hemispherical, the r1 and r2 will be same. So the pressure will be delta P equal to sigma by r. Then you can see, as I was saying, that once the hole is being formed, it is governed by the scalar quick relation, which is V equal to square root of 2 sigma. This rho W is the water density only, and H is the hemispherical cap height. So the refraction velocity of the hole, how fast the hole is being propagated to the bubble, that is given by this relation. So you can see that depending upon the surface tension, the Taylor quick relation is essentially saying that if we add surfactants, the whole propagation will be slow. Because surfactant it reduces the like surface tension of the fluid. Now H is the bubble thickness. So what happens here is the experimental observation. What it says that the bubbles, as it lasts longer and longer, due to this, like as I said, due to the gravity, the upper portion essentially kind of drains out the fluid towards the bottom side. The fluid, like the bubble, becomes thinner. So it is said that the older bubbles which are more thinner emit more smaller and faster droplets when it is bursting out compared to a younger bubble. Younger bubbles are more thicker. So these are all like experimental studies. On the other hand, there is another like competing phenomena, that is like let's say there is something called melancholy flow. Do you know the melancholy flow? Melancholy flow happens due to the surface tension gradient. So like let's say there is a higher surface tension and lower surface tension. The lower surface tension fluid will be basically pulled by the higher surface tension region. Okay. So now let's say there is some surface tension gradient here, like let's say some cold air is being like here blowing over, or due to evaporation there is cooling happening. So at lower temperature we know that in case of water, surface tension increases with water. So if there is some sort of localized cooling here, then there will be increasing sigma compared to the ground level. So there is a surface tension gradient. So in the last slide I said that gravity essentially tries to drain out the fluid from the top portion to the bottom portion. Here, due to the magnetic flow, the reverse is happening. From the lower region, the fluid is trying to going towards the higher surface tension region. So that is basically like a reverse mechanism which will like what I can say that will increase the duration of the bubble to stay. Essentially, like as the bubble becomes thinner, it has high tendency to burst out, right? So this melancholy flow is like opposing that mechanism. So the melancholy flow can happen due to temperature gradient, like bubbles emerging into cooler atmospheres. Like as I said, if like let's say some sort of localized cool air is blowing, then evaporative cooling, some sort of evaporation is taking place, then it is being like released, then there is cooling. Maybe there are chemical or biological compounds which is reducing the surface tension. That is also again leading to the variation in the surface tension. And sometimes it has been seen bacteria they can secrete some biopolymers which will lead to a change in the surface tension of the base fluid. That can also help in this sort of melancholy flow. Next one are some general slides I just made long before. These are the like other motivations of studying evaporation. This can be like let's say from a healthy individual to a like a person who is sick. If we study the drying pattern of the evaporation, like the evaporating blood droplets, you can I'm not going to the details, but the drying pattern of a healthy individual and a sick individual, depending upon like let's say someone is suffering from some blood related diseases, the drying pattern will be different in them. So Professor Saptaji Vasu is studying those sort of things, many French cases are also studying. Then here the evaporation will also depend upon the what kind of surface, whether it is hydrophilic, whether it is super hydrophobic. The hydrophilic one will have more contact area, the super hydrophobic will have minimal contact area. So depending upon that also the evaporation rate is also dependent. Recently I saw in the can you give me water in terms of water? So many of you have seen the David Goodin's the drop paper. He came in come to IIT Hyderabad. So he was studying the how to say like the static splashing dynamics of blood. Like if someone hits you bullet from whichever directions, not you, it's like some poor victim. Blood, like the drying pattern of the blood splash, he have done like they have collected from different directions. So basically, what he did, he took many of his like lab students to collected the blood sample and he is like impacting that in different directions, studying the patterns. Then he has collected this database. This he has used as a like some sort of data model and he used AIML techniques to launch a app for the city where he stays. He is working with that municipality or their police, like the forensic service. Basically, he has launched an app with which you can take the photo of the splashing figure, the dried up splashing figure of that blood drop. and then he can help in the forensic analysis from which angle the bullet has been like hit, or like how the whether it is a blunt weapon or a sharp weapon, that sort of like some sort of insight into the criminal activities that he can, like at least he is claiming so. He has been working in that direction. So some like this I just this you can see like in a hydrophilic flow at the edges here will be the highest like highest amount of loads and rate. So what happens like this figure came in the context of office and read. Many times like there is this paper you can read later in a much more detail like DWGN Nature I think 2000 paper. So this paper is just type coffee stain or coffee ring effect. What happens if we leave the coffee to dry? It forms a circular stain. So what happens essentially they are saying like at the edges the evaporation rate is highest. So fluids from these edges they are traveling to the outer periphery. So let's say a coffee drop, the whole fluid is essentially covered with coffee particles. Since at this surrounding the circle, evaporation rate is highest, the coffee particles are essentially going rapidly outward and they accumulate in that this outer periphery and that leads to the drying up of the coffee particles leading like the ultimate coffee ring is formed. So there like subsequently there also have been studied how to suppress this coffee ring. Like what I would suggest at least first you can read the coffee ring effect. That is a very interesting 2 3 page paper only. So now, just like briefly, I will say the modes of evaporation. One mode of evaporation is called the constant radius mode. Essentially, you are seeing here that if you look, this is the initial droplet and the configuration. It gets pinned at one place and then it goes down. So you can see here from highest to lowest. Essentially, it means the volume is gradually reducing. However, since these points are fixed, you can see the radius is like constant. On the other hand, if you measure the contact angle is like the horizontal line and the periphery of these droplets, you can see the contact angle is decreasing. So this is called the constant radius mode. Likewise, the other mode is called constant angle mode. You can see here that it is actually gradually reducing gradually. So the radius is decreasing here, its radius is decreasing, but it is like a self-similar profile. The contact angles in most cases are constant. So you can see here the constant, like it is called the constant contact angle mode. Sorry, just this one, like the third one is called the hybrid, like it can also, like it can be a mixture of two cases, that is, it is called six side mode. There is like a constant radius mode you can see, and then the radius mode is falling. Correspondingly, this constant radius, you can see the contact angle is increasing, and the second part, the contact angle is constant. So these are the typical modes which we normally encounter in evaporation. Then the last part contains the disease transmission in plants, where essentially you can see a drop falling on the horizontal surface creates some sort of crashing like this, whereas the leaves are generally hung like this, and leaves are also flexible surfaces. So when the droplet falls down like this, the leaf may also oscillate, and that may also lead to greater like transmission of the droplets when it breaks it. So here you can see this is like let's say what if the leaf is just horizontal line. But what if the leaf is bent and it also simultaneously interact with the impacting drop? Okay. So that is again some sort of fluid structure interaction problem. It can be like bungless and some other BTech people they started doing this experiment on a simple cantilever beam. Like obviously they are not as soft as the beams, but nevertheless those beams are flexible when it like impacts a drop. It basically in some cases it fragments and spreads out, and in some cases it sticks to the surface. But that is again depending upon the droplet impact velocity. As well as like we are doing in polymers, the polymer concentration will also come into the picture. So here you can see that these are basically like after the fragmentation, what is the map of the pathogen footprint. So you can see here is a pathogen footprint when, like let's say there is a water film or a sessile drop. Just a simple water droplet is here. So water, how the fragmentation is creating the map of the dispersed droplet. So you can see here the different surfaces. This is for inclined drop, rigid surface, how it is creating here. The compliant surface means it's a flexible surface actively interacting with the impacting drop. So you can see in all the three cases, the footprint of the dispersed droplets are different. Then these are the different sorts of, like some in some cases it is called crescent moon fragmentation for a drop or drop impact. So instead of water film, if there is some sort of sessile droplet already reached, you can see it's like can be imagined as a half moon. That's why it's called crescent moon. So here also there is, like you can see that let's say this leaf is some sort of, as we like interact with the drop, the leaf is also interacting. So you can see this is like, let's say if this leaf there is drop impacting, then the leaf may also like some sort of oscillation, you can create oscillation and it can create the rebound of the droplets of the impacting droplet. So these are with water droplets. In some cases, when the non-nutritional like contribution is sufficient enough, even though the leaf oscillates, it can do this long filament. This is water here. The filament is breaking up. In our case, if we add like let's say 1500 or 2000 ppm, this filament will not break. It will stay with the leaf. Okay, so this is the final or whatever. I don't know how many slides are still there. Okay, just one slide. So okay, so these are like finally like the fragmentation state where it is like impacting on the surface. The general parameters you can see the seed evolution when you are basically forming the governing equations. Obviously, the velocity you have to put, how the thickness profile. Expected it is difficult, but it is not impossible to measure. People having sufficient like some other advanced measurement techniques, or I don't know how they means maybe the lens is powerful enough to magnify along the side views, then they can measure the thickness and the rim instability can also be studied. How many, like let's say for a given amount of the rim diameter, how many thin filament structures are appearing and how the fragmentation process is taking place. So these sort of study are also being done in the context of how the fragmentation leads to more and more pathogen transmission. So I've discussed in brief about like several aspects, drop impact dynamics, splash dynamics, as well as like evaporation dynamics. And let's say in case of plant surfaces, drop impact on flexible surfaces. So I also have tried to connect with how our lab is trying to like explore this sort of dynamics, and we showed that like some times like the suppression of splashing, those things can be minimized by adding little bit of polymer in the fluid. So these are the summary, or like let's say these are the important hydrodynamic aspects which we try to cover briefly. That's all for this one. Then switch off. Now maybe questions or maybe questions in the team. Done.